Hello, and welcome to my presentation on glycolysis and gluconeogenesis for Chemistry 530 at Grand Canyon University. My name is Drew Sword, and I'll be taking you through this presentation. For this presentation, we'll first start with an overview of both pathways for glycolysis and gluconeogenesis. We'll then take a look at aldolase involvement. We'll then take a look at isoforms and expression patterns of aldolase. We'll take a closer look at aldolase B expression. And finally, we'll have our conclusion. On the next slides, we will discuss the pathways of both glycolysis and gluconeogenesis. Glycolysis is a metabolic pathway occurring in the cytosol of the cell that is the starting reaction of cellular respiration and anaerobic respiration. In glycolysis, glucose is broken down into two pyruvate molecules in a 10-step process with two phases, an investment phase that consumes 2 ATP for energy, and a payoff stage that produces 4 ATP, which nets 2 ATP for energy use. The pyruvate molecules can then go on to react in the citric acid cycle to form 32 more ATP in the aerobic cellular respiration, or are turned into lactate through anaerobic respiration, forming 2 ATP. Also produced in glycolysis are 2 NADH molecules, which can also lead to more ATP production for the body. The steps of glycolysis, according to Khan Academy, are: Step 1, a phosphate group is transferred from ATP to glucose, making glucose 6-phosphate. Glucose 6-phosphate is more reactive than glucose, and the addition of the phosphate also traps glucose inside the cell, since glucose with a phosphate can't readily cross the membrane. Step 2, glucose 6-phosphate is converted into its isomer, fructose 6-phosphate. Step 3, a phosphate group is transferred from ATP to fructose 6-phosphate, producing fructose 1,6-bisphosphate. This step is catalyzed by the enzyme phosphofructokinase, which can be regulated to speed up or slow down the glycolysis pathway. Step 4, fructose 1,6-bisphosphate splits to form 2,3-carbon sugars, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate. They are isomers of one another, but only one, the glyceraldehyde 3-phosphate, can directly continue through the next steps of glycolysis. Step 5, the DHAP is converted into glyceraldehyde 3-phosphate, and the two molecules exist in equilibrium, but the equilibrium is pulled strongly downward in the scheme of the diagram above, or to the left, as glyceraldehyde 3-phosphate is used up. Thus, all of the DHAP is eventually converted. Two half reactions in step 6 occur simultaneously. The first, glyceraldehyde 3-phosphate, is oxidized, and 2-NAD+ is reduced to NADH and H+. The overall reaction is exergonic, releasing energy that is then used to phosphorylate the molecule, forming 1,3-bisphosphoglycerate. Step 7, 1,3-bisphosphoglycerate donates one of its phosphates groups to ADP, making a molecule of ATP and turning into 3-phosphoglycerate in the process. Step 8, 3-phosphoglycerate is converted into its isomer, 2-phosphoglycerate. Step 9, 2-phosphoglycerate loses a molecule of water, becoming phosphoenol pyruvate, which is known as PEP. PEP is an unstable molecule poised to lose its phosphate group in the final step of glycolysis. And then step 10, PEP readily donates its phosphate group to ADP, making a second molecule of ATP. As it loses its phosphate, PEP is converted to pyruvate, the end product of glycolysis. Gluconeogenesis is much like glycolysis, only the process occurs in reverse. However, there are exceptions. In glycolysis, there are 3 highly energetic steps, steps 1, 3, and 10. These are also regulatory steps, which include the enzymes hexokinase, phosphofructokinase, and pyruvate kinase. Biological reactions can occur in both the forward and reverse direction. If the reaction occurs in the reverse direction, the energy normally released in that reaction is now required. If gluconeogenesis were to simply occur in reverse, the reaction would require too much energy to be profitable to that particular organism. In order to overcome this problem, nature has evolved 3 other enzymes to replace the glycolysis enzymes hexokinase, phosphofructokinase, and pyruvate kinase mentioned earlier when going through the process of gluconeogenesis. The first step in gluconeogenesis is the conversion of pyruvate to phosphophenyl pyruvic acid, PEP. In order to convert pyruvate to PEP, there are several steps and several enzymes required. Pyruvate carboxylase, PEP carboxykinase, and malate dehydrogenase are the 3 enzymes responsible for this conversion. Pyruvate carboxylase is found on the mitochondria and converts pyruvate into oxaloacetate. Because oxaloacetate cannot pass through the mitochondria membranes, it must be first converted into malate by malate dehydrogenase. Malate can then cross the mitochondria membrane into the cytoplasm, where it is then converted back into oxaloacetate with another malate dehydrogenase. Lastly, oxaloacetate is converted into PEP via PEP carboxykinase. The next several steps are exactly the same as glycolysis, only the process is in reverse. The second step that differs from glycolysis is the conversion of fructose 1,6-bisphenol phosphate to fructose 6-phenyl phosphate with the use of enzyme fructose 1,6-phosphatase. This conversion of fructose 6-phosphate to glucose 6-phosphate uses the same enzyme as glycolysis, phosphoglucoisomerase. The last step that differs from glycolysis is the conversion of that glucose 6 to the glucose with the enzyme glucose 6-phosphatase. This enzyme is located in the endoplasmic reticulum. In the next part of this presentation, we will take a closer look at step 4 in glycolysis and step 7 in gluconeogenesis, the utilization of the enzyme aldolase. In glycolysis, fructose 1,6-bisphosphate aldolase catalyzes the reversible aldol cleavage of fructose 1,6-bisphosphate and fructose 1-phosphate to dihydroxyacetone phosphate, DHAP, and either glyceraldehyde 3-phosphate, GAP, or glyceraldehyde. DHAP and GAP are constitutional isomers. Only GAP can continue on in the glycolysis process, so another enzyme, triose phosphate isomerase, converts DHAP into GAP. From this point on, there are 2 molecules processed in each step, 5 through 10, of glycolysis. In the gluconeogenesis process, aldolase from step 7 converts DHAP and GAP into fructose 1,6-bisphosphate. It is important to note that glycerol phosphate from triglyceride catabolism is converted into DHAP for the condensation with GAP, forming D-fructose 1,6-bisphosphate. There are 3 different isoforms of aldolase: aldolase A, aldolase B, and aldolase C, which each have expressions in different tissues within the body. In the next slides, the 3 isoforms will be discussed as well as their expressions. There is also a closer look into aldolase B and its expression regulation. Aldolase A, like its other isoforms, is a key enzyme in glycolysis and gluconeogenesis, especially because it is expressed in most tissues, though it is predominantly expressed in developing embryo and adult muscle. Aldolase A also contributes to other functions, including muscle maintenance, regulation of cell shape and motility, striated muscle contraction, actin cytoskeleton organization, and regulation of cell proliferation. Aldolase A is highly expressed in multiple cancers, including lung squamous cell carcinoma, renal cancer, and hepatocellular carcinoma. It is proposed that aldolase A overexpression enhances glycolysis in these tumor cells, promoting their growth. While aldolase A and C are important isoforms, aldolase B, which is found in the liver, kidneys, and small intestine, is the primary isoform in fructose metabolism, and most of the fructose is metabolized in these locations. Genetic mutations of aldolase B cause hereditary fructose intolerance, which causes a backup of fructose 1-phosphate and keeping phosphate from being released, resulting in the cessation of glycogenolysis in the liver, causing hypoglycemia. We will take a closer look at aldolase B in a future slide. Aldolase C, while also important to glycolysis and gluconeogenesis, has a very specific function different than the other isoforms, as it participates in the stress response pathway for lung epithelial cell function during hypoxia. Aldolase C is most commonly found in brain and neuronal tissue and is also in the central nervous system. As discussed in previous slides, all 3 isoforms of aldolase are involved in the glycolysis and gluconeogenesis pathways. Aldolase A is most prevalent in muscle tissue and red blood cells. Aldolase B is primarily found in the liver, kidneys, and small intestine. And aldolase C is primarily found in the brain and neuronal tissues. In taking a closer look at aldolase B for its expression regulation, there seem to be 2 binding sites for aldolase B activation: the C/EBP-DBP binding site and the HNF1 and HNF3 overlapping binding site. Both DBP and HNF1 are strong transactivators of the aldolase B promoter, with the C/EBP being a weak activator. HNF3 is an anti-activator, completely antagonizing transactivation by HNF1 and non-competitively antagonizing transactivation by DBP. Both the glycolysis and gluconeogenesis pathways play important roles in aerobic and anaerobic respiration. Both pathways allow for ATP to be produced to provide energy for the body, and the aldolase isoforms play important roles in both pathways in different tissues throughout the body. Thank you so much for listening to my presentation on glycolysis and gluconeogenesis. And here is my information. And here are the references that I used.